Okay, as we begin this biochemistry lecture, just a few notes on the web page for AP Biology. I have posted the lecture slides for you to jot some things down as we move along. And also there is a reading on there regarding chirality with isomers. And it's an evolutionary aside. Evolution is an embedded concept within each chapter as an underlying theme for our biology studies. So take a moment to read that article as well. So we ended with this the other day. We said that we were looking at dehydration synthesis, hydrolysis reactions, and we know that in a dehydration synthesis reaction, you have the formation of a larger molecule by the removal of water. So basically, you're going to have monomers that are going to come together to form the larger molecules called polymers. The reverse is a hydrolysis reaction, and in a hydrolysis reaction, water is going to serve as a reactant. And hydrolysis, so you're going to split water, hydro meaning water, lysis is to split. You're going to split water, and water is going to be used to break down the larger polymer molecules essentially into their monomer subunits. So here you can see a small example of what those reactions would look like. In letter A here we have a dehydration synthesis reaction, and in letter B we have, of course, the hydrolysis reaction. So our first group is the carbohydrates. Carbohydrates are the sugars. These are molecules that have a 1 to 1 ratio of carbon, hydrogen, to oxygen. So we say that their empirical formula is CH2ON. And whatever integer would fall in N, you just distribute that through to figure out what the formula would be for that particular sugar you're talking about. When you look at carbohydrates are essentially molecules that are composed of carbon and water. And we do see that here, carbon and water, in the empirical formula. That bond between carbon and hydrogen is a covalent bond that holds much energy. Carbohydrates are a good energy storage molecule. We could get some of that energy out of basic sugars, starches, and glucose itself, which is blood sugar. So when we go to look at the different types of carbohydrates, we're going to look at those carbohydrates that we consider monosaccharides. Then there are disaccharides and polysaccharides. So let's take a look at monosaccharides first. Monosaccharides are the simplest carbohydrate form. So monosaccharides are going to serve as the subunit, the monomer subunits to the larger molecules. You have those monosaccharides that contain 6 carbons, which are our hexose sugars. Really quick, I will put that in there. This you should remember from regular biology, advanced biology. So 6 carbon sugars are called hexose sugars. And those hexose sugars, an example would be glucose. Glucose is the blood sugar at about 0.5%. Its empirical formula is C6H12O6, so there's where we see those 6 carbons. There are also 2 other hexose sugars, or monosaccharides. That would be fructose, and fructose is a structural isomer of glucose. And also you have galactose, and galactose is a stereoisomer of glucose. And we went over the difference between stereoisomers and structural isomers. So what we see there is glucose itself is blood sugar in animals. Fructose is what we would call fruit sugar in plants. And galactose is a component of milk sugar. And when you look at the different structural and stereoisomer formulas, you know that enzymes, or not formula, structures, it's going to be enzymes that are going to act on those different sugars that can distinguish the structural and stereoisomers of the basic 6-carbon skeleton. So here you can see basically this is the straight chain form of a 6-carbon sugar. That would be glucose. So here it is in straight chain form. Here you can see the 6-carbon backbone, and off of there, of course, we have our functional groups. Both are hydroxyls, and then down here we have that carbonyl. Also, what you see is a ring form. So here would be the ring form structure of glucose. And when you go into ring form, you could have an alpha glucose or a beta glucose. This is a little bit more in depth than what you've learned sophomore year, but basically you're looking at where that hydroxyl group is attached. So in an alpha glucose, you could see where it is attached on the first carbon of that carbon ring. And down here in the beta glucose, you could see where it's attached there in the first carbon of the carbon ring. So here it is down, and here it is in an upward position off of that carbon. Looking again at the differences between glucose, galactose, and fructose, here you can see glucose itself in its straight chain form. As far as structural isomer, you could see where it's different than right here. And then a stereoisomer. Remember, stereoisomers give you that mirror image. So here you see that mirror image here with galactose. So when we look at the building of these larger sugars from the monosaccharide subunits, first we're going to look at when two join together. Two monosaccharides joining together equals a disaccharide. So two monosaccharides linked via dehydration synthesis forms a disaccharide. The bond between two sugars is called a glycosidic linkage. The glycosidic linkage. Basically, disaccharides are used for sugar transport or energy storage. And examples would be sucrose, lactose, and maltose. These would be transport form or storage forms of sugar in plants and animals. So if you look down below, you do see the formation of sucrose via a dehydration synthesis reaction. And basically, we would have alpha glucose forming with fructose. And you're going to remove that hydroxyl group from fructose and the hydrogen off of the hydroxyl group from glucose. That will give us water, so those molecules have been dehydrated, and now you have a spot there to form a bond. And that is going to bring glucose plus fructose is going to equal sucrose there. Sucrose itself is common table sugar. It's the transport form of sugar in plants. The one we see over here is maltose. Maltose is often found in animals, and that is when you have glucose bonding with another glucose molecule. So that would be a dehydration synthesis reaction involving two glucose molecules. And then, of course, lactose is milk sugar. Often humans lack the enzyme, a sufficient amount of the enzyme lactase to break down milk sugar into its individual components, and that of course would eventually lead to lactose intolerance. And if you are lactose intolerant, you know a lot of the complications, abdominal complications, stomach complications that come along with being lactose intolerant. But lactose, when the enzyme lactase works on breaking down that disaccharide lactose, the two monomer subunits there to form lactose would be glucose and galactose. And then, of course, you would have the polysaccharides. Polysaccharides are long chains of monosaccharides that are linked through dehydration synthesis. A lot of these are used for energy storage, such as plants. The energy storage polysaccharide there would be starch. Starch is stored in the plants. In the animals, the storage form of that carbohydrate or polysaccharide would be glycogen. And glycogen, of course, is stored in the liver. Then you also have structural polysaccharides. Structural polysaccharides would include cellulose cellulose is the major component of all plant cell walls. Cellulose is one of the most abundant organic compounds on Earth because it is found in plant cell walls as that rigid structure that gives it that cell wall its rigidity, unlike the cell membrane that all cells have. Also, you have the structural polysaccharide called chitin. Remember in biology that CH gives you a K sound. So there we have chitin, and chitin is a structural polysaccharide found in the exoskeleton of arthropods, and it is also a structural polysaccharide found in fungi. If you look at them itself, there you have that linkage forming those right here. You see that glycosidic linkage that is used to bring together those glucose molecules. So if you look down here, you can see glycogen is a long chain that is highly branched. That's storage form of carbs in the animals. And if you look here, here we see the storage form of carbohydrates in plants themselves. Our next group, of course, would be the nucleic acids. Nucleic acids is the group that's often forgotten. Nucleic acids, the polymer itself is called a nucleic acid. This would be DNA, RNA. The monomers that make nucleic acids are called nucleotides. And basically we know that those monomers are sugars plus a phosphate and a nitrogenous base. The sugar in DNA is deoxyribose. In RNA, that sugar is ribose. These sugars are monosaccharide sugars. They are 5-carbon sugars that we call pentose sugars. And then, of course, you have the nitrogenous bases, along with that functional group, a phosphate. The nitrogenous bases, there are 2 types. There are those nitrogenous bases that are purines, and then there are those that are pyrimidines. You can remember the nitrogenous bases. I always think of adenine and guanine, A and G, kind of like agriculture, and you always want to have pure agriculture. So purines are the adenine and guanine, and then the pyrimidine nitrogenous bases would be thymine, cytosine, and uracil. Nucleotides are then connected, if you have that long backbone, are then connected by the phosphodiester bonds. So those are the bonds that are going to bring those nucleotides together, the phosphodiester bonds. If you look at the structure of a nucleotide itself, here's where you'll have one of those nitrogenous bases attached to a primary carbon in that pentose ring sugar, whether it be RNA or DNA. So this sugar could be either deoxyribose or ribose. And then here's that functional group that we find in nucleic acids called the phosphate. And then that itself, here we see that long chain, and here's where we see that phosphodiester bond. So you can see here the actual structure of each nucleotide. So adenine and guanine, cytosine is both in DNA and RNA, thymine is a nucleotide found in DNA only, and uracil is only found in RNA. Deoxyribonucleic acid encodes information for amino acid sequencing in proteins. And we'll look at this in greater detail once we get into protein synthesis. And I'm going to stop there for now. This will conclude our first online lecture for biochemistry. If you have any questions, please jot them down, and we will answer them in class. Thank you and have a nice day.